Hello YouTube, my name is Neil and we're studying for the MCATs today. We're in the Biochemistry Review. Chapter 1, Amino Acids, Peptides, and Proteins. How important are amino acids? Consider sickle cell disease. People with sickle cell disease have red blood cells that under certain conditions can be rigid and sickle-shaped. Those sickle-shaped cells can become stuck in capillaries blocking them. In severe cases, it can block enough of the blood supply to cause damage to several organs, such as the kidneys, liver, and spleen. This happens because of a mutation in hemoglobin, the protein in red blood cells that transports oxygen. Remarkably, the difference between the normal form of hemoglobin, HbA, and the one that causes sickle cell disease, HbS, is a seemingly minor one. All it takes is a change in a single amino acid on the surface of hemoglobin. The sixth amino acid in two of its four chains is changed from glutamic acid to valine. This minor difference allows the deoxygenated form of HbS to aggregate and precipitate and leads to the sickle shape and all of the symptoms of sickle cell disease. In this chapter, we'll take a look at the basics of proteins by focusing on the amino acids that compose them and how those amino acids contribute to the physical and chemical properties of proteins. (1.1) Amino acids found in proteins. Amino acids are molecules that contain two functional groups, an amino group, the NH2, and a carboxyl group, COOH. In this chapter, we'll focus specifically on the alpha amino acids, in which the amino acids in the carboxyl group are bonded to the same carbon. The alpha carbon of the carboxylic acid. Think of the alpha carbon as the central carbon of the amino acid, as shown in figure 11 or 1.1. And we have a basic amino acid. I'm sure this is that glutamine? That's not glutamine. What is the most basic amino acid? Glycine. Is this glycine? This has an R group. Glycine doesn't even have an R group. The alpha carbon in glycine so this would be glycine if R was just a hydrogen. In addition to the amino and carboxyl groups, the alpha carbon has two other groups attached to it, a hydrogen atom and a side chain, also called an R group, which is specific to each amino acid. The side chains determines the properties of amino acids and therefore their functions. So let's draw the basic amino acid structure. We have an H bonded to an N, bonded to another H, bonded to our alpha carbon, which is bonded to an R group, which could just be a hydrogen. And then a COOH, or an OOH. Yeah, a COOH, a carboxyl group. Should we do it in blue? It's because they did theirs in blue. A note on terminology. Amino acids don't need to have both the amino and carboxyl groups bonded to the same carbon. For example, the neurotransmitter gamma aminobutyric acid, GABA, has the amino group on the gamma carbon, 3 carbons away from the carboxyl group. Similarly, not every amino acid found in the human body is specified by a codon in the genetic code or incorporated into proteins. One example is ornithine. Of the intermediates in the urea cycle, the metabolic process by which the body excretes excess nitrogen. There are also some amino acids that are specifically modified for specialized roles in the body. For example, lysine is sometimes converted into pyrolysine, and cysteine is sometimes converted into selenocysteine. That said, the Association of American Medical Colleges, AAMC, has specifically stated they'll focus on the 20 alpha amino acids encoded by the human genetic code, also called proteinogenic amino acids. So for the rest of this chapter, we'll use the term amino acid to refer specifically to these compounds. So there are possible amino acids that are not, how do they put it? Alpha amino acids, the 20 alpha amino acids encoded by the human genetic code, also called proteinogenic. Proteinogenic amino acids are the 20 alpha amino acids encoded by genome, human genome. And an example of a gamma amino acid or a gamma amino butyric acid, also known as GABA, Stereochemistry of amino acids. For most amino acids, the alpha carbon is a chiral or stereogenic center, as it has 4 different groups attached to it. Thus, most amino acids are optically active. The one exception is glycine, which has a hydrogen atom at its R group, making it achiral. So let us draw glycine. You got the carboxyl, and since one of the R's is a hydrogen, instead of drawing the R group down, they actually draw the nitrogen down because it's more interesting. But I kind of want it similar to the others. I want the hydrogen in the R group spot, and I want nitrogen up here. It is achiral, so I guess either of those conformations is possible. Nitrogen, nitrogen, and another hydrogen. I mean, I guess I'm not really specifying whether they're going into the page or out of the page here. But if we were to make a, what's that diagram called? The Fisher projection. And they do go on to make Fisher projections in a minute. So we'll take a look at those. But this is glycine. All chiral amino acids used in eukaryotes are L amino acids. So the amino group is drawn on the left in a Fisher projection as demonstrated in figure 1.3. In the Kahn-Ingold pre-log system, this translates to an S-absolute configuration for almost all chiral amino acids. The only exception is cysteine, which, while still being an L-amino acid, has an R-absolute configuration because the CH2SH group has priority over the COOH group. The sulfur, the highly substituted carbon gets priority over the carboxyl carbon. They have a picture of it. They do not. But let's draw rather an L and D amino acid. COOH on top, R on the left, H, NH2, and COOH, R on the bottom, NH2 on the right, and H on the left. The NH2 on the left would be our L amino acid, and the NH2 on the right would be our D amino acid. Traditionally L, or for most of them, L amino acids are also S, but it is not in the case of cysteine, which is R. Traditional, these are going to be R. Except cysteine, which is R. All right. Structures of the amino acid. There are several ways to classify amino acids. In this section, we'll break them down by the structure of their side chains. We'll start with nonpolar, non-aromatic side chains. We did a quick review of these in organic, but now we're going to go and draw each one down to get a better understanding, I guess. And there's abbreviations. All right, we'll get into that. Nonpolar, non-aromatic. Seven amino acids shown in figure 1.4a fall into this class. Glycine, discussed earlier, has a single hydrogen atom as its side chain and is therefore achiral. It is also the smallest amino acid. Four other amino acids, alanine, valine, leucine, and isoleucine, have alkyl side chains containing 1 to 4 carbons. Methionine is one of only 2 amino acids that contains a sulfur atom in its side chain. Nevertheless, because the sulfur has a methyl group attached, it is considered relatively nonpolar. Finally, proline is unique in that it forms a cyclic amino acid. In all the other amino acids, the amino group is attached only to the alpha carbon. In proline, however, the amino nitrogen becomes a part of the side chain, forming a 5-membered ring. That ring places notable constraints on the flexibility of proline, which limits where it can appear in a protein and can have significant effects on proline's role in secondary structure. Alanine, valine, leucine, isoleucine, all alkyl side chains. 1, 3, 1, 2, 3, 4, 1, 2, 3, 4, and 4 carbons. Methionine with the sulfur, and then an alkyl group after that. And proline, where its side chain makes a ring with the nitrogen, so the nitrogen is actually part of the side chain. Let's draw these real quick. Did we already do glycine? We did do glycine. We forgot H2. But in these pictures, they have the side chains going off to the left, and they have the amino group going down. So I'm going to move this and change it. I'm going to grab this. And we're going to do this to it. I'll put the NH2. H. That's glycine. Alanine. Has just a simple 1-carbon alkyl group. Valine has an isopropyl group. Leucine has 1, 2, 3, 4. A butyl, I guess isobutyl group. Isoleucine looks like leucine, except instead of the isobutyl, it's got a methyl group and a second methyl group. 1, 2, 3, 4. Still a butyl R group, a 4-carbon R group. Methionine's got 1, 2 carbons, a sulfur, and then another carbon in its R group. And proline with the ring structure. And instead of an NH2, it's just an NH. That didn't look too good. Let's do that again. Let's draw the pentagon first. Let's do that again. There we go. We got proline, methionine, isoleucine, leucine, valine, and alanine in reverse order, of course, to how I drew them. Slow this down a little bit. All right. Aromatic side chains next. The 3 amino acids have uncharged aromatic side chains. And are depicted in figure 1.4b. The largest of these is tryptophan, which has a double ring system that contains a nitrogen atom. The smallest is phenylalanine, which has a benzyl side chain, benzene ring plus a CH2 group. Adding an OH group to phenylalanine gives the third member, which is tyrosine, which has a hydroxyl group on the ring. While phenylalanine is relatively nonpolar, the hydroxyl on the tyrosine makes it polar. And they didn't say explicitly, but tryptophan doesn't look too polar down on this end, does it? It's got a nitrogen. Is nitrogen does hold electrons a little, doesn't it? Never mind. Tyrosine or tryptophan probably is charged or polar. Is tryptopolar. Tryptophan is considered nonpolar. Just tyrosine's polar here. Let's draw these out. Tryptophan, double ring, 1,6 carbon ring, followed by a 5 carbon, one in which a nitrogen replaces the carbon, so actually a 4 carbon, 1 nitrogen ring, but a pentagon. They don't draw it like a pentagon, but you should, right? Something like this. Got a double bond here. And we have the rest of the amino acid. We have phenylalanine, also 6 carbon ring, an extra carbon attaching it to the rest of the amino acid. Finally, tyrosine, which looks pretty similar to phenylalanine, but it has an extra hydroxyl group on the ring, making it polar. Tryptophan, phenylalanine, and tyrosine. Polar side chains. Five amino acids shown in figure 1.4c have side chains that are polar but not aromatic. Serine and threonine Both have hydroxyl groups on their side chains, which make them highly polar and able to participate in hydrogen bonding. Asparagine and glutamine have amide side chains, unlike the amino group common to all amino acids. The amide nitrogens do not gain or lose protons with changes in pH. They do not become charged. What's the difference between amine and amide? Amine. Amide. So amides are amines that specifically are attached to alpha carbons. A carbon that's carboxyl carbon. And what's the difference between carboxyl and carbonyl again? Carboxyl is COOH. Carbonyl is just a double bond between a carbon and an oxygen. So an amide is bonded to a carbonyl carbon, while an amine is just a nitrogen anywhere. And they're saying that amides so in this case, the amino nitrogen is the amide. It is bonded to the alpha carbon, right? In all the ones that we're studying. Unlike the amino group common to all amino acids, the amide nitrogens do not gain or lose protons with changes in pH. So what are the amide nitrogens? Let's take a look at them. So the nitrogens at the alpha carbon is not what they're talking about. They're talking about this other nitrogen down here. It's on a carbonyl carbon, but not our carboxyl carbon. Also, glutamine has an amide. It's got a nitrogen bonded to our carbonyl. And those are the only ones with nitrogen, other than our main amino group. Let's continue reading. Unlike the amino group common to all amino acids, the amide nitrogens do not gain or lose protons with changes in pH. They do not become charged. The last amino acid with a polar side chain is cysteine, which has a thiol or a sulfur hydrogen group on its side chain. Because the sulfur is larger and less electronegative than oxygen, the SH bond is weaker than the OH bond. This leaves the thiol group in cysteine prone to oxidation, a reaction we'll study later in this chapter. All right, cysteine gets oxidized. Which means losing bonds to hydrogen and gaining bonds to oxygen. Or gaining electrons. No, gaining electrons is being reduced. Being oxidized is losing electrons. So losing bonds to oxygen and gaining hydrogen bonds. So it'll pick up a hydrogen. Let's draw these few amino acids, starting with serine, which has, after our carboxyl and amino, one carbon and then a hydroxyl. Threonine, which has a carbon with a hydroxyl and an alkyl group on it as an R chain, as a side chain. We have asparagine, which has a carbon, a carbonyl carbon, meaning connected to an oxygen, double bonded to an oxygen, followed by nitrogen. Carbon, carbonyl carbon, nitrogen. Glutamine, which looks similar to asparagine with an extra carbon. So 1, 2 carbons, and then a carbonyl, and then the amide. One, two. And then finally cysteine, which has just a carbon and a sulfur for its side chain. Carbon, sulfur, SH. Serine, 3O. You know what? I need more room. Because when I try to grab these, it'll grab the words. So let's do this first. Serine, threonine, asparagine, glutamine, and cysteine. Negatively charged or acidic side chains. Only 2 of the 20 amino acids have negative charges on their side chains at physiological pH of 7.4. Those 2 are aspartic acid, aspartate, which is related to asparagine, and glutamic acid, glutamate, which is related to glutamine. Unlike asparagine and glutamine, aspartate and glutamate have carboxylate, COO instead of COOH groups in their side chains rather than amides. Note that aspartate is simply the deprotonated form of aspartic acid, and glutamate is the deprotonated form of glutamic acid. These 2 amino acids are depicted in figure 1.4d. So we got aspartic acid and glutamic acid, and then we have aspartate, which is the anion, where it loses its carboxyl hydrogen. So maybe we don't have to draw the ions, but we'll just write aspartic acid and glutamic acid. All right. Positively charged side chains. The remaining 3 amino acids shown in figure 1.4e have side chains that have positively charged nitrogen atoms. Lysine has a terminal primary amino. Terminal primary amino group, while arginine has 3 nitrogen atoms in its side chain. The positive charge is delocalized over all 3 nitrogen atoms. The final amino acid, histidine, has an aromatic ring with 2 nitrogen atoms. This ring is called imidazole. You might be wondering how histidine can acquire a positive charge. The pKa of the side chain is relatively close to 7.4. It's about 6. So at physiological pH, one nitrogen atom is protonated and the other isn't. Under more acidic conditions, the second nitrogen atom can become protonated, giving the side chain a positive charge. Let's draw these complicated amino acids. Starting with arginine, which has 3 nitrogens in the side chain. And 4 carbons. We have lysine, which has just 1 nitrogen in its side chain. Whoops. H2N. 1, 2, 3, 4, and 4 carbons in the side chain alone. Finally, histidine, which has 2 nitrogens in a ring structure in its side chain. A couple double bonds. 1, 2, and then an extra 2 carbons to link it to the rest of the amino acid. Looks pretty good. Histidine, preceded by lysine, preceded by arginine. All right. All right. These are all positively charged. And I guess the nitrogen was the one that was a little confusing. But it can become more protonated, which is when it becomes positively charged. So it's only under acidic conditions that this will become positively charged, I guess. Hydrophobic and hydrophilic amino acids. Classifying amino acid side chains as hydrophobic or hydrophilic is actually a very complex matter. For example, tyrosine has both a hydroxyl group and an aromatic ring. So which one wins? A few clear conclusions can be drawn, though. First, the amino acid with long alkyl side chains, alanine, isoleucine, leucine, valine, and phenylalanine, are all strongly hydrophobic and thus more likely to be found in the interior of proteins, away from water on the surface of the protein. Second, all the amino acids with charged side chains, positively charged histidine, arginine, and lysine, plus negatively charged glutamate and aspartate, are hydrophilic, as are the amides, asparagine, and glutamine. The remaining amino acids lie somewhere in the middle and are neither particularly hydrophilic nor particularly hydrophobic. So why don't we draw this out a little bit? Let's put hydrophobic on the left, hydrophilic on the right. So I'll make a little arrow. Put hydrophobic on the left, hydrophobic, hydrophilic. And let's put alanine, isoleucine, leucine, valine, phenylalanine. Let's put all those guys over there on the left in the hydrophobic side. Charged side chains: histidine, arginine, lysine, glutamate, aspartate, are all hydrophilic. And the rest are somewhere in between. All right. Amino acid abbreviations. Now that we have explored the structures of the 20 proteinogenic amino acids, it is worth mentioning that you are expected to be able to identify an amino acid on the MCAT not only by name, but also by its 3-letter and 1-letter abbreviations. Alanine, ALA, arginine, ARG, asparagine, ASN, aspartic acid, ASP, cysteine, CYS, glutamic acid, GLU, glutamine, GLN, glycine, GLY, histidine, HIS, isoleucine, ILE, leucine, LEU, lysine, LYS, methionine, MET, phenylalanine, PHE, proline, PRO, serine, SER, threonine, THR, tryptophan, TRP, tyrosine, TYR, valine, VAL. Those are your 3-letter abbreviations. And then the 1-letter abbreviations, alanine is A, arginine is R, asparagine is N, aspartic acid is D, cysteine is C, glutamic acid is E, glutamine is Q. Interesting. Glycine is G, histidine is H, isoleucine is I, leucine is L, lysine is K, methionine is M, phenylalanine is F, proline is P, serine is S, threonine is T, tryptophan is W, tyrosine is Y, and valine is V. It's nice that we got a little chart, but we're going to have to become familiar enough to basically have this memorized. All right, some questions. What are the 4 groups attached to the central alpha carbon of a proteinogenic amino acid? The 4 groups attached to the alpha carbon are, of course, there's the carboxyl group, there's the amino group, there's 1 hydrogen, and there's 1 R group. That R group can be a hydrogen. It could be an alkyl group. Could be a ring. It's going to be a carbon, but it could be a carbon attached to a nitrogen. That R group always starts with a carbon, except for in the case of cysteine or glycine. Nitrogen, carbon. Right, but it could be a heavily substituted alkyl group, either with hydroxyls, with amides, with just carbons, with sulfur. All right. What is the stereochemistry of the chiral amino acids that appear in eukaryotic proteins? All the eukaryotic proteins are L. What do they call it? They're all L amino acids, which makes them all, incidentally, mostly S, except for cysteine, which is R. So the R or S, they're all S except for cysteine, which is R. But cysteine is still an L amino acid. They're all L amino acids. Which amino acid fits into each of these categories? Note the number in parentheses indicates the number of amino acids in this category. So non-polar, non-aromatic. I mean, we could just go through them. Glycine, alanine, valine, leucine, isoleucine, methionine, proline. Polar. Serine, threonine, asparagine, glutamine, cysteine. Negatively charged. Aspartic acid, glutamic acid. Positively charged. Arginine, lysine, histidine. Did we say the aromatics? We said polar. Did I skip aromatics? Tryptophan, phenylalanine, and tyrosine. Number 4, where do hydrophobic amino acids tend to reside within a protein? What about hydrophilic ones? Hydrophobic would be on the interior of the protein. Hydrophilic would be on the exterior where it's exposed to the water. Should we check the answers to those? Pretty confident about that. The 4 groups are an amino group, a carboxylic acid group, a hydrogen atom, and an R group. Number 2, all chiral eukaryotic amino acids are L. All chiral eukaryotic amino acids are S, with the exception of cysteine, which is R. 3. Nonpolar non-aromatics are glycine, alanine, valine, leucine, isoleucine, methionine, proline. The aromatics are tryptophan, phenylalanine, tyrosine. The polar side chains are serine, threonine, asparagine, glutamine, and cysteine. I said those. Negatively charged, aspartate, glutamate. Positively charged, lysine, arginine, histidine. And number 4, hydrophobic amino acids tend to reside in the interior of a protein, away from water. Hydrophilic amino acids tend to remain on the surface of the protein in contact with water. Great. On to 1.2 after I take a quick bathroom break. All right. 1.2, acid-base chemistry of amino acids. Acid-base chemistry. The AAMC loves to use amino acids to test your understanding of acid-base chemistry because they have both an acidic carboxylic acid group and a basic amino group. That makes them amphoteric species that can either accept a proton or donate a proton. How they react depends on the pH of their environment. The key to understanding the behavior of amino acids is to remember 2 facts. 1. Ionizable groups tend to gain protons under acidic conditions and lose them under basic conditions. So in general, at low pH, ionizable groups tend to be protonated. At high pH, they tend to be deprotonated. The pKa, number 2, the pKa of a group is the pH at which, on average, half of the molecules of that species are deprotonated. That is, protonated version of the ionizable group equals deprotonated version of the ionizable group, or HA, which equals A. The number of base equals the number of acid. So half deprotonated. If the pH is less than pKa, a majority of the species will be protonated. If the pH is higher than the pKa, a majority of the species will be deprotonated. Right on. Protonation and deprotonation. Should I write these down? Just remember that at pKa, half the ions are protonated. Protonation and deprotonation. Because all amino acids have at least 2 groups that can be deprotonated, they all have at least 2 pKa values. The first one, pKa1, is the pKa for the carboxyl group and is usually around 2. For most amino acids, the second pKa value, pKa2, is the pKa for the amino group, which is usually between 9 and 10. For amino acids with an ionizable side chain, there will be 3 pKa values, but we'll come back to that later. As an example, let's take glycine, which doesn't have an ionizable side chain. So let's write down that pKa1 is around 2, pKa2 is around 9, between 9 and 10. pKa1 So that's a hard thing to write down. PK sub A sub 1. pKa1. Is for the COOH, the carboxyl group. And it's around 2. PK sub A sub 2 is for the amino group, and it's between 9 and 10. Positively charged under acidic conditions at pH 1, below even the pH of the stomach. There are plenty of protons in solution because we're far below the pKa of the amino group. The amino group will be fully protonated. So the amino group will be fully protonated to NH3 and thus positively charged. Because we're also below the pKa of the carboxylic acid, it too will be fully protonated, COOH, and thus neutral. Therefore, at very acidic pH values, amino acids tend to be positively charged, as shown in figure 1.5. And we have a basic amino acid with just an R for the R group, for the side chain. And the NH3, the nitrogen is fully protonated, as well as the carboxyl carbon, or the carboxylate group. The OH on the carboxyl carbon has its hydrogen. It's fully protonated. So at physiological pH, usually this OH is not protonated, while this is, at around 7, and that's what we'll get into in a minute, I guess. So under acidic conditions, the nitrogen is fully protonated and the hydroxyl group is protonated. Let's put a little plus on there. And they actually draw coming into and out of the page, so let's do that. Going into the page, coming out of the page. Let's just turn this into blue for good measure, because that's what they do, how they have it. All right. Zwitter ions at intermediate pH. If we increase the pH of the amino acid solution from 1 to 7.4, the normal pH of human blood, we've moved far above the pKa of the carboxylic acid group. At physiological pH, you will not find amino acids with the carboxylate group protonated, the COOH, and the amino group unprotonated, NH2. Under these conditions, the carboxyl group will be in its conjugate base form and be deprotonated, becoming COO-. Conversely, we're still well below the pKa of the basic amino group, so it will remain fully protonated and in its conjugate acid form, NH3+. Thus, we have a molecule that has both a positive charge and a negative charge, but overall the molecule is equally neutral, electrically neutral. We call such molecules dipolar ions or Zwitter ions, from the German Zwitter or hybrid. As depicted in figure 1.6, the two charges neutralize one another, and Zwitter ions exist in water as internal salts. Internal salts, great. acidic conditions, let's write down physiological conditions, which is what we mean by intermediate or neutral. Nah, let's not do that actually. Just neutral conditions. You have our O minus instead of COOH, but our nitrogen is still fully protonated. Giving it that plus charge. And then at negatively charged basic conditions, milk of magnesia, which is often used as an antacid, has a pH around 10.5. At that pH, the carboxylate group is already deprotonated and thus remains COO minus. On the other hand, we are now well above the pKa for the amino group, so it deprotonates as well, becoming NH2. So at highly basic pH, glycine is now negatively charged, as depicted in figure 1.7. So we're going to predict what that looks like. O minus, R group, nitrogen with just 2 hydrogens on it, and a negative charge or a neutral charge. Let's go with neutral charge. Let's see what they do. Neutral. Totally, like in its total charge, its overall charge is minus. All right. Titration of amino acids. Because of these acid-base properties, amino acids are great candidates for titrations. We assume that the titration of each proton occurs as a distinct step, resembling that of a simple monoprotic acid. Thus, the titration curve looks like a combination of 2 monoprotic acid titration curves or 3 curves if the side chain is charged. Figure 1.8 shows the titration curve for glycine. After we inspect this curve, we'll look at the differences for the amino acids with charged side chains. So we have a pKa around 2.3, another pKa around 9.6, and between the two we have a a halfway point or a midpoint of 5.9. Of course, that first pKa is for the amino group. Second pKa is for the carboxylic acid. Imagine an acidic 1 mole glycine solution. At low pH value, glycine exists predominantly in NH3CH2COOH, fully protonated. It is fully protonated with a positive charge. As the solution is titrated with NaOH, the carboxyl group will deprotonate first because it is more acidic than the amino group. When half the equivalent of base has been added to the solution, the concentrations of the fully protonated glycine and its zwitterion, NH3 plus CH2COO minus, are equal. That is, the amount of protonated and deprotonated carboxylic acid at this point is equal. At this point, the pH equals pKa1. Remember, when the pH is close to the pKa value of a solute, a solution is acting as a buffer, and the titration curve is relatively flat, as demonstrated in the blue box in the diagram. As we add more base, the carboxylate group goes from half deprotonated to fully deprotonated. The amino acid stops acting like a buffer, and pH starts to increase rapidly during this phase. When we've added one equivalent of base, glycine exists exclusively as the Zwitter-ion form. Remember, we started with one equivalent of glycine. This means that every molecule is now electrically neutral, and thus the pH equals the isoelectric point P1, or Pi rather, of glycine. This is true for all amino acids. The isoelectric point is the pH at which the molecule is electrically neutral. For neutral amino acids, it can be calculated by averaging the two pKa values for the amino and carboxyl groups. Just a simple average. pKa1 plus pKa2 divided by 2. For glycine, the Pi value is 2.34 plus 9.60 divided by 2, which comes to 5.97. Remember that when the molecule is neutral, it is especially sensitive to pH changes, and the titration curve is nearly vertical. As we continue adding base, glycine passes through a second buffering phase as the amino group deprotonates. Again, the pH remains relatively constant. When 1.5 equivalents of base has been added, the concentration of the Zwitter-ion form equals the concentration of the Zwitter-ion form equals the concentration of the fully deprotonated form. That is, the NH3 plus COO minus equals NH2 COO minus. And the pH equals pKa2. Once again, this titration curve is nearly horizontal. Finally, when we've added two equivalents of base, the amino acid has become fully deprotonated, and all that remains is NH2COO minus. Additional base will only increase the pH further. Should we write down an equation for the isoelectric point? We could do that. It's pretty simple, but it might help to have it written down or look at it visually. Pi of neutral amino acid equals pKa1 plus pKa2 all over 2, average. Amino acids with charged side chains. For amino acids with charged side chains, such as glutamic acid and lysine, the titration curve has an extra step, but works along the same principles as described above. Because glutamic acid has two carboxyl groups and one amino group, its charge in its fully protonated state is still +1. It undergoes the first deprotonation, losing the proton from its main carboxyl group, just as glycine does. At that point, it is electrically neutral. When it loses its second proton, just as with glycine, its overall charge will be -1. However, the second proton that is removed in this case comes from the side chain carboxyl group, not the amino group. This is a relatively acidic group with a pKa of around 4.2. The result is that Pi of glutamic acid is much lower than that of glycine, around 3.2. The isoelectric point for an acidic amino acid can be calculated as follows. Pi equals pKa of the R group plus pKa of the carboxylic acid group divided by 2. So similar to this equation, except instead of the two pKa's of the amino and carboxylic acid, there's a third pKa, the pKa of the R group. And now we're taking the average of pKa1 and the R group instead of pKa2. Lysine, on the other hand, has two amino groups and one carboxyl group. Thus, its charge in its fully protonated state is +2, not +1. Losing the carboxyl proton, which still happens around pH 2, brings the charge down to 1. Lysine does not become electrically neutral until it loses the proton from its main amino group, which happens around a pH of 9. It gets a negative charge when it loses the proton on the amino group and its side chain, which happens around pH 10.5. Thus, the isoelectric point of lysine is the average of the pKa values for the amino group and side chain. The pi is around 9.75. So the isoelectric point for a basic amino acid can be calculated as follows. The pi equals the pKa of the R group plus the pKa of the amino group divided by 2. The take-home message: amino acids with acidic side chains have relatively low isoelectric points, while those with basic side chains have relatively high electric points. I like that summary. Let's write that down. Acidic amino acids have low pi values, and basic amino acids have high pi values. All right. Some questions. For a generic amino acid, NH2CrHCOOH, with an uncharged side chain, what would be the predominant form at each of the following pH values? At a pH of 1, it'll be fully protonated, NH3 and COOH. At a pH of 7, its COOH will be deprotonated, but its NH3 will still be protonated. If it's at this point, if its side chain is uncharged, its overall charge will also be neutral. At a pH of 11, the NH3 also becomes deprotonated, the amino group also becomes protonated, and this kind of amino acid would be negatively charged at this point. Given the following pKa values, what is the value of pi for each of the following amino acids listed? Aspartic acid, an acidic amino acid with a pKa 1 of 1.8, a pKa 2 of 3, and a pKa 3 of 9. Its isoelectric point would be between its first two pKa values, ignoring the third. And so its pi would be around 2. We could throw this into our calculator to get a good number, a good exact number. We'll take 1.88 plus 3.65 divided by 2. 2.765. Should we write that down? Let's write that down. We skipped 1. We're just going straight. Or we didn't skip 1. We did it. We just didn't write it down. 2 we're going to write down because we're not going to remember these values. Aspartic acid has a pi of 2.765. Arginine, pKa 1 of 2.7, pKa 2 of 9.4, pKa 3 of 12.48. So it's basic. It's got to be between the 9.04 and 12.48. 9.04 plus 12.48 divided by 2. 10.76. Finally, valine, which only has 2 pKa values, so it's going to be right between the two, 2.32 plus 9.62 divided by 2, 5.97. Let's Let's write aspartate, arginine, and valine. And let's check those answers real quick. (1.2) pH at a pH of 1, it's NH3COOH. At 7, it's NH3COO minus. And at pH of 11, it's NH2COO minus. Number 2, aspartic acid has a pI of 2.77, arginine 10.76, and valine 5.97. Great. On to 1.3. Peptide bond formation and hydrolysis. Hydrolysis. Peptides are composed of amino acid subunits, sometimes called residues, as shown in figure 1.9. Dipeptides consist of 2 amino acid residues. Tripeptides have 3. The term oligopeptide is used for relatively small peptides up to 20 residues, while longer chains are called polypeptides. Let's write that down. Oligopeptide is less than 20 residues, and a polypeptide is greater than. So we just put polypeptide over here. The residues in peptides are joined together through peptide bonds, a specialized form of an amide bond that forms between the COO minus group of one amino acid and the NH3 plus group of another amino acid. This forms a functional group, CONH. In this section, we'll look at the key reactions involved in forming and breaking peptide bonds. So the peptide is specifically CONH. Peptide bond formation. Peptide bond formation is an example of a condensation or dehydration reaction because it results hydrolysis. Hydrolysis is the breaking of peptide bonds. Dehydration or condensation is the peptide bond formation. So let's remove hydrolysis and put dehydration instead. Condensation / dehydration. Peptide bond formation is an example of condensation or dehydration because it results in the removal of a water molecule, H2O. It can also be viewed as an acyl substitution reaction, which can occur with all carboxylic acid derivatives. When a peptide bond forms, as shown in figure 110, the electrophilic carbonyl carbon on the first amino acid is attacked by the nucleophilic amino group on the second amino acid. After that attack, the hydroxyl group of the carboxylic acid is kicked off. The result is the formation of a peptide or amide bond. So to compare to this, CO minus, that O minus becomes OH, and that OH gets kicked off as water. Because amide groups have delocalizable pi electrons in the carbonyl and in the lone pair on the amino nitrogen, they can exhibit resonance. Thus, the CN, the carbon-nitrogen bond in the amide, has partial double bond character as shown in figure 111. We have this double bond on the oxygen in resonance with a double bond on the nitrogen. As a result, rotation of the protein backbone around the carbon-nitrogen amide bond is restricted, which makes the protein more rigid. Rotation around the remaining bonds in the backbone, however, is not restricted, as those remain single pi bonds. When a peptide bond forms, the free amino end is known as the amino terminus, or N-terminus, while the free carboxyl end is the carboxy terminus, or C-terminus. By convention, peptides are drawn from the N-terminus on the left and the C-terminus on the right. Similarly, they are read from N-terminus to C-terminus. N on the left, C on the right. Let's draw the resonance of the peptide bond, and then we'll also label the N and C ends, the nitrogen and carbon ends. We're going to draw it with the nitrogen to the left, we're going to do it with its 3D structure. NH, and then another R group of our next amino acid, and the O, the COO minus, kind of upside down in this case. And its resonance looks pretty similar, but a single bond to a negatively charged oxygen and then a double bond to a positively charged nitrogen. And our second R group and our other carboxylic acid. And of course, our N-terminus and our C-terminus. Not between these structures, but there's an N and a C on each of these. Let's put them together on the same molecule. How about that? N-terminus, C-terminus. All right. Peptide bond hydrolysis. For enzymes to carry out their function, peptides need to be relatively stable in solution. Therefore, they don't normally fall apart on their own. On the other hand, in order to digest proteins, we need to break them down into their component amino acids. In organic chemistry, amides can be hydrolyzed using acid or base catalysis. In living organisms, however, hydrolysis is catalyzed by the hydrolytic enzymes such as trypsin and chymotrypsin. Both are specific in that they only cleave at specific points in the peptide chain. Trypsin cleaves at the carboxyl end of arginine and lysine, while chymotrypsin cleaves at the carboxyl end of phenylalanine, tryptophan, and tyrosine. While you don't need to know the exact mechanism of how these enzymes catalyze hydrolysis, you do need to understand the main idea. They break apart the amide bond by adding a hydrogen atom at the amide nitrogen and an OH hydroxyl group at the carbonyl carbon. This is a reverse reaction shown in figure 110. So let's write down trypsin, spelled correctly. Trypsin. They both cleave at the carboxyl end of arginine and lysine, at the carboxyl end of phenylalanine, tryptophan, and tyrosine. Trypsin for arginine, and lysine, chymotrypsin cleaves phenylalanine, tryptophan, and tyrosine. Just going to turn these into blue. It's kind of like that convention. All right. What is the difference between an amino acid, a dipeptide, a tripeptide, an oligopeptide, and a polypeptide? An amino acid is just one peptide. A dipeptide is 2 amino acids. A tripeptide is 3. Oligo is up to 20, and polypeptide is more than 20. What molecule is released during formation of a peptide bond? Water, because it is a dehydration reaction. If chymotrypsin cleaves at the carboxyl end of phenylalanine, tryptophan, and tyrosine, how many oligopeptides would be formed in enzymatic cleavage of the following molecule of chymotrypsin? Phenylalanine, tryptophan, tyrosine. Phenylalanine, tryptophan, tyrosine. So we'd have one here. So we'd have 2 pieces after that. Tyrosine, 3 pieces after that, and then another on trip, 4 pieces, and then another on tyrosine again. 5, 6 pieces. 1. Let's double check that we have the abbreviations right. We're looking for phenylalanine, tryptophan, and tyrosine. Phenylalanine, PHE, tryptophan, TRP, and tyrosine, TYR. TRP and TYR. PHE, TYR, TRP, TYR. 1, 2, 3, 4, resulting in 5 pieces. How many oligopeptides? Well, the first on the far end of phenylalanine will be a dipeptide, not an oligopeptide. The second, 1, 2, 3, will be a tripeptide, not an oligopeptide. The third, just from tyrosine to phenylalanine, will just be a single peptide, monopeptide. And then again, after tryptophan, a monopeptide. And then we have 1, 2, 3, a tripeptide. And then a dipeptide. We will not have any oligopeptides. They'll all be tris and dis, but we'll have 1, 2, 3, 4, 5, 6 pieces. None of them oligopeptides. Let's see what they say. 1.3, number 1. These species differ by the number of amino acids that make them up. Amino acid has 1, dipeptide has 2, tripeptide has 3, oligopeptide has few, less than 20, polypeptide has many, more than 20. So they do call these Oh no, we'll get into that. Number 2, water leaves as H2O in the formation of a peptide bond. And number 3, we have the valine phenylalanine piece. We have the glutamine, lysine, tyrosine piece. We have the, what is ILE? Isoleucine, methionine, tyrosine piece, glycine, alanine. A single amino acid on its own is not considered an oligopeptide. So they have 1, 4. 1, 2, 3, 4. They have the dipeptides and tripeptides included. 1, 2, 3, 4. And they excluded the, for example, the tryptophan piece. So 1, 2, the phenylalanine piece and the tryptophan piece. But they do have a third and a fourth. All right, so they considered dipeptides and tripeptides oligopeptides, but not single amino acids. So they came up with 4. All right. Oligopeptides are small, less than 20, but more than 1. At least dipeptides. All right. 1.4, primary structure and secondary protein structure. Proteins are polypeptides that range from just a few amino acids in length up to thousands. They serve many functions in biological systems, functioning as enzymes, hormones, membrane pores, and receptors, and elements of cell structure. Proteins are the main actors in cells. The genetic code, after all, is simply a recipe for making thousands of proteins. Proteins have 4 levels of structure: primary, secondary, tertiary, and quaternary. In this section, we'll examine the first 2. We'll discuss tertiary and quaternary structure in the next section. So let's just write down structure. Primary structure of a protein is the linear arrangement of amino acids coded in the organism's DNA. It's the sequence of amino acids listed from the N-terminus or amino end to the C-terminus or carboxyl end. So, for example, the first 10 amino acids of the beta chain of hemoglobin are normally valine, histidine, leucine, threonine, proline, glutamate, 2 glutamates, lysine, serine, and alanine. Primary structure is stabilized by the formation of covalent peptide bonds between adjacent amino acids. The primary structure alone encodes all the information needed for folding at all of the higher structural levels, the secondary, tertiary, and quaternary structures. A protein adopts the secondary, tertiary, and quaternary structures a protein adopts are the most energetically favorable arrangements of the primary structure in a given environment. The primary structure of a protein can be determined by a laboratory technique called sequencing. This is the most easily done using the DNA that coded for that protein, although it can also be done from the protein itself. Primary structure is literally the genetic code. Sequence of residues. Secondary structure. Secondary structure. A protein's secondary structure is the local structure of neighboring amino acids. Secondary structures are primarily the result of hydrogen bonding between nearby amino acids. The 2 most common secondary structures are alpha helices and beta-pleated sheets. The key to the stability of both structures is the formation of intramolecular hydrogen bonds between different residues. Hydrogen bonds between neighboring residues. Hydrogen bonds between neighboring residues, which results in alpha helices and beta pleated sheets. All right. Alpha helices. The alpha helix, shown in figure 1.12, is a rod-like structure in which the peptide chain coils clockwise around a central axis. The helix is stabilized by intramolecular hydrogen bonds between a carbonyl oxygen atom and an amide hydrogen atom, 4 residues down the chain. The helix is stabilized by intramolecular hydrogens between a carbonyl oxygen atom and an amide hydrogen atom, 4 residues down the chain. The side chains of the amino acids in the alpha helical conformation point away from the helix core. The alpha helix is an important component in the structure of keratin, a fibrous structural protein found in human skin, hair, and fingernails. So the side chains point away from the center, from the core. It's found in keratin. Well, let's write some of these attributes to alpha helices. Hydrogen bond between the carbonyl oxygen is bonded to amide hydrogen. And in the peptide bond, the amide hydrogen is singular, just one hydrogen. Kicks off 2 hydrogens. Four residues down. This picture isn't great. I'd like to take a look at like a 3D picture of that. Because they show you the hydrogen bond with the dots between, I guess this would be a carbonyl oxygen and this would be the amide hydrogen. And so 1, 2, 3, 4 peptides down. The carbonyl hydrogen bonded to a hydrogen, 1, 2, 3, 4 down. 4 peptides down the chain is what they're getting at. Let's also write down that it's in keratin. Side chains point away from the center. We'll see if that's important. Beta-pleated sheets. In beta-pleated sheets, which can be parallel or anti-parallel, the peptide chains lie alongside one another, forming rows or strands held together by intramolecular hydrogen bonds between carbonyl oxygen atoms on one chain and amide hydrogen atoms in an adjacent chain. As shown in figure 113, to accommodate as many hydrogen bonds as possible, the beta-pleated sheets assume a pleated or rippled shape. The R groups of amino residues point above and below the plate of the beta-pleated sheet. Fibroin, the primary protein component of silk fibers, is composed of beta-pleated sheets. So let's just write that beta-pleated sheets make up fibrin. And it's also a hydrogen bond between a carboxyl oxygen and an amide hydrogen. So this applies to both of them. So I'm going to put this down under here, and this just for both of these. All right. Secondary structure of proline. Because of its rigid cyclic structure, proline will introduce a kink in the peptide chain when it is found in the middle of an alpha helix. Proline residues are thus rarely found in alpha helices, except in helices that cross the cell membrane. Similarly, it is rarely found in the middle of pleated sheets. On the other hand, proline is often found in the turns between the chains of a beta-pleated sheet and is often found as the residue at the start of an alpha helix. So let's just write down that proline makes a kink. Proline is associated with a kink in either an alpha helix or a beta-pleated sheet. In the beta-pleated sheet, that kink might result in the fold and either at the beginning and I guess at the beginning of an alpha helix. Proline makes a kink. Great. You're not going to find it in the middle of a structure. You might find it at the end. Some questions. What are the definitions of primary and secondary structure, and how do they differ in subtypes, and the bonds that stabilize them? Well, the primary structure is literally the linear sequence of amino acids. So subtypes, they're all peptide bonds. They're always going to be a carbonyl attached to an amide. Beyond that, the different types, each of the peptides could have a slightly different bond, but subtypes, I'd say no, they're all peptide bonds. Stabilizing bonds. What stabilizes a peptide bond? The resonance between the carbonyl and the amide. Secondary structure is how neighboring residues interact with each other, sometimes making hydrogen bonds. So the stabilizing bond would be a hydrogen bond. The subtypes, you have alpha helices and beta-pleated sheets. What role does proline serve in secondary structure? Well, it makes a kink, so it's not going to be found in the middle of either an alpha helix or beta-pleated sheet, but you might find it at the folds of the beta-pleated sheet or at the beginning of an alpha helix. Let's check those. 1.4, primary structure, linear sequence of amino acids interchange. Subtypes, none. Stabilizing bonds, the peptide bond. Secondary structure, local structure determined by nearby amino acids, interactions with the neighbors. Subtypes, alpha helix and beta pleated sheet, and the stabilizing bond is the hydrogen bond. Proline's rigid structure causes it to introduce kinks in alpha helices or creates turns in beta pleated sheets. So kinks in helices, turns is for beta pleated sheets. Kink/turn. All right. Next, tertiary and quaternary protein structure. Primary, secondary, tertiary. Proteins can be broadly divided into fibrous proteins, such as collagen, that have structures that resemble sheets or long strands, and globular proteins, such as myoglobin, that tend to be spherical, that is, like a globe. These are caused by tertiary and quaternary protein structures, both of which are the result of protein folding. Tertiary. Fibrous. Sheets or long strands like collagen. And globular, like myoglobin. Tertiary structure. A protein's tertiary structure is its three-dimensional shape. Tertiary structures are mostly determined by hydrophilic and hydrophobic interactions between R groups of amino acids. Hydrophobic residues prefer to be on the interior of proteins, which reduces their proximity to water. Hydrophilic, nitrogen-hydrogen and carbon-oxygen double bonds found in the polypeptide chain get pulled in by these hydrophobic residues. Hydrophilic bonds get pulled in by hydrophobic residues. These hydrophilic bonds can then form electrostatic interactions and hydrogen bonds that further stabilize the protein from the inside. As a result of these hydrophobic interactions, most of the amino acids on the surface of proteins have hydrophilic, polar, or charged R groups. Highly hydrophobic R groups, such as phenylalanine, are almost never found on the surface of a protein. The three-dimensional structure can also be determined by hydrogen bonding, as well as acid-base interactions between amino acids with charged R groups, creating salt bridges. A particularly important component of tertiary structure is the presence of disulfide bonds, the bonds that form when two cysteine molecules become oxidized to form cysteine. Cysteine. Two cysteine molecules become oxidized to form cysteine, as shown in figure 114. Disulfide bonds create loops in the protein chain. In addition, disulfide bonds determine how wavy or curly human hair is. The more disulfide bonds, the curlier it is. Note that forming a disulfide bond requires the loss of 2 protons and 2 electrons, oxidation. Disulfide bond, the product of oxidation, makes curls in hair as well as loops in proteins at large. Is cysteine important? Should we draw cysteine to cysteine? I think it's just enough to know that disulfide bonds are responsible for loops. So, tertiary structure is influenced by hydrophobic / hydrophilic interactions, as well as hydrogen bonding, as well as acid-base interactions, and in particular, disulfide bonds. Disulfide bonds make loops and is an example of oxidation. The exact details of protein folding are beyond the scope of the MCAT, but the basic idea is that the secondary structures probably form first, and then hydrophobic interactions and hydrogen bonds cause the protein to collapse into the proper three-dimensional structure. Along the way, it adopts intermediate states known as molten globules. Protein folding is an extremely rapid process from start to finish. It typically takes much less than a second. If a protein loses its tertiary structure, a process commonly called denaturation or denaturation, it loses its function. So misfolded proteins are problems. Misfolded proteins do not work correctly. A lot of diseases are due to misfolded proteins, isn't it? Folding and the solvation layer. Why do hydrophobic residues tend to occupy the interior of a protein, while hydrophilic residues tend to accumulate on the exterior portions? The answer can be summed up in one word: entropy. Whenever a solute dissolves in a solvent, the nearby solvent molecules form a solvation layer around the solute. From an enthalpy standpoint, even hydrocarbons are more stable in aqueous solution than in organic ones. However, when a hydrophobic side chain, such as those in phenylalanine and leucine, is placed in aqueous solution, the water molecules in the solvation layer cannot form hydrogen bonds with the side chain. This forces the nearby water molecules to rearrange themselves into specific arrangements to maximize hydrogen bonding, which means a negative change in entropy. Remember that negative changes in entropy represent increasing order, decreasing order, decreasing disorder, and thus are unfavorable. This entropy change makes the overall process non-spontaneous. On the other hand, putting hydrophilic residues such as serine and lysine on the exterior of the protein allows the nearby water molecules more latitude in their positioning, thus increasing their entropy and making the overall solvation process spontaneous. Thus, by moving hydrophobic residues away from the water and molecules and hydrophilic residues toward the water molecules, a protein achieves maximum stability. Quaternary structure. Just wanted to move this over a little bit to align it a little bit better. Quaternary structure. All proteins have elements of primary, secondary, and tertiary structure. Not all proteins have quaternary structure. Quaternary structure only exists for proteins that contain more than one polypeptide chain. For these proteins, the quaternary structure is an aggregate of smaller globular peptides, or subunits, and represents the functional form of the protein. The classic example of quaternary structure are hemoglobin and immunoglobulins, shown in figure 1.15a and b. Hemoglobin consists of 4 distinct subunits, each of which can bind 1 molecule of oxygen. Similarly, immunoglobulin G, IgG, antibodies also contain a total of 4 subunits each. The formation of quaternary structures can serve several roles. First, they can be more stable by further reducing the surface area of the protein complex. Second, they can reduce the amount of DNA needed to encode the protein complex. Third, they can bring catalytic sites close together, allowing intermediates from one reaction to be directly shuttled to a second reaction. Finally, the most important, they can induce cooperativity or allosteric effects. We'll discuss much further in the next chapter, especially for hemoglobin, but the basic idea is that one subunit can undergo conformational or structural changes which either enhance or reduce the activity of the other subunits. Quaternary structure refers to the subunit arrangement. Contain more than one polypeptide chain. Multiple chains The quaternary structure can further stabilize the molecule, can reduce the amount of DNA needed to encode the protein. It could bring catalytic sites together. Allosteric effects. Allosteric effects. Cooperativity. All right. Conjugated proteins. Conjugated proteins derive part of their function from covalently attached molecules called prosthetic groups. These prosthetic groups can be organic molecules such as vitamins or even metal ions such as iron. Proteins with lipid, carbohydrate, and nucleic acid prosthetic groups are referred to as lipoproteins, glycoproteins, and nucleoproteins, respectively. These prosthetic groups have major roles in determining the function of their respective proteins. For example, each of the hemoglobin subunits, as well as myoglobin, contain a prosthetic group called heme. The heme group, which contains an iron atom in its core, binds to and carries oxygen. As such, hemoglobin is inactive without the heme group. These groups can also direct the protein to be delivered to a certain location, such as the cell membrane, nucleus, lysosome, or endoplasmic reticulum. So a lipoprotein is going to have a lipid part and a protein part, giving it some functionality in both those realms, giving it some water solubility as well as some fat solubility. And glycoproteins are going to be some combination of sugar and protein. Nucleoproteins, I guess, are going to be have a nucleic acid group as well as a protein group to get into the nucleus of the cell and have some kind of functionality there. Let's write down conjugated proteins. Have prosthetic groups and some examples. Vitamins, metal ions, iron. Proteins with lipid, carbohydrate, and nucleic acid prosthetic groups. Let's write each of those down. lipoproteins have lipids. Glycoproteins have carbohydrate. Prosthetic groups. And nucleoproteins have nucleic acid prosthetic groups. Great. Heme is an important one. Some questions. What are the definitions of tertiary and quaternary structure, and how do they differ in subtypes and the bonds that stabilize them? The definition of tertiary structure is a good question. Strict definition is its three-dimensional shape, which is, of course, influenced by hydrophobic, hydrophilic interactions, hydrogen bonds, acid-base interactions, as well as disulfide bonds. Those are all the stabilizing interactions. Subtypes, fibrous or globular. Quaternary structure, that would be its subunit arrangement if it has multiple polypeptide chains. Subtypes, I guess there's a lot of them. But I don't think there's an answer for that, because each protein is going to have like a unique type based on this level of structure. So I don't know if there's subtypes. We'll see the answers in a minute. But stabilizing bonds are going to be these are going to be in complexes that are going to be brought together by unique proteins. So I would say that their stabilizing bonds are also unique. So maybe no answer across the board for this. We'll see what they say in a minute. What is the primary motivation for hydrophobic residues in a polypeptide to move to the interior of the protein? What is the motivation for hydrophobic residues in a polypeptide to move to the interior of the protein? They're more stable, meaning they have lower energy requirements to put them into that configuration, and that's because interactions with water, right? Or hydrophobic / hydrophilic interactions. So to increase entropy. So I guess the ultimate motivation is entropy. Increase their entropy. Let's see what their answers are. 1.5. Tertiary structure, three-dimensional shape of the protein. Subtypes, hydrophobic interaction, acid-base or salt bridges, as well as disulfide links. They left out hydrogen bonding, but that's one as well. Oh, the stabilizing bonds are hydrogen bonds, ionic bonds, covalent bonds, and van der Waals forces. So I didn't write all those down. There are all the types of bonds will be involved in this level of structure. Even van der Waals forces. Quaternary structure. Do you remember what van der Waals forces are? I think it's when nearby molecules kind of adopt the charge or the orientation of their neighbors, and you kind of get a cumulative effect. What's van der Waals again? Van der Waals. Relatively weak electric forces that attract neutral molecules to one another in gases, in liquefied and solidified gases, and in almost all organic liquids and solids. So weak electric forces that are attractive, generally. Quaternary structure. The definition is the interaction between separate subunits of a multi-subunit protein. Subtypes, there are none. Stabilizing bonds, same as tertiary structure. All of the bonds are involved. Covalent, ionic, hydrogen, they're all involved in this level of structure. Number 2, moving hydrophobic residues to the interior of a protein increases entropy by allowing water molecules on the surface of the protein to have more possible positions and configurations. This positive delta S makes delta G less than 0, stabilizing the protein. Great. Increasing entropy. 1.6, denaturation. Denaturation. How do you pronounce? Denaturation.
Denaturation. Denaturation.
They say denaturation. I like denaturation. Naturation? Okay, naturation. In the previous section, we discussed protein folding. The reverse of this process is denaturation, in which a protein loses its three-dimensional structure. Although it is sometimes reversible, denaturation is often irreversible. Whether its denaturation is reversible or not, unfolded proteins cannot catalyze reactions. The two main causes of denaturation are heat and solutes. As with all molecules, when the temperature of a protein increases, its average kinetic energy increases. When the temperature gets high enough, this extra energy can be enough to overcome the hydrophobic interactions that hold a protein together, causing the protein to unfold. This is what happens when egg whites are cooked. In the uncooked egg whites, albumin is folded, which makes it transparent. Cooking then causes the albumin molecules to denature and aggregate, forming a solid, rubbery white mass that will not revert to its transparent form. On the other hand, solutes such as urea denature proteins by directly interfering with the forces that hold the protein together. They can disrupt tertiary and quaternary structures by breaking disulfide bridges, reducing cysteine back to 2-cysteine residues. They can even overcome the hydrogen bonds and other side chain interactions that hold alpha helices and beta pleated sheets intact. Similarly, detergents such as SDS, sodium dodecyl sulfate, also called sodium lauryl sulfate, can solubilize proteins, disrupting non-covalent bonds and promoting denaturation. Denaturation. Sometimes reversible, caused by heat or solutes. In the solute case, let's note SDS, sodium dodecyl sulfate or sodium lauryl sulfate. I think we use that in lab quite a bit as a buffer, right? SDS page. All right. Why are proteins denatured by heat and solutes, respectively? Well, heat, extra energy, allows hydrophobic regions to overcome their their fear of water, and so they're able to unfold. While in the solute case, our solutes are actually interfering with the bonds, like the disulfide bond, breaking them, causing their structure to unfold similarly. Let's see what they say real quick. Heat denatures proteins by increasing their average kinetic energy, thus disrupting hydrophobic interactions. Solutes denature proteins by disrupting elements of secondary, tertiary, and quaternary structure. the bonding elements specifically of secondary, tertiary, and quaternary structure. Great. On to practice questions. Number 1. In a neutral solution, most amino acids exist as positively charged compounds, zwitterions, negatively charged compounds, or hydrophobic molecules. I think the answer's got to be zwitterions. We could go back and check real quick. Zwitterions. We call such my... I'm pretty sure it's zwitterions. We'll go with the zwitterions. 2. At pH 7, the charge on a glutamic acid molecule is... Well, it's the acid, so it's naturally acidic. It will be a zwitterion. It'll be neutral. It'll be isoelectric, rather. It'll be isoelectric between a low pH and another low pH. At 7, it'll be well above its isoelectric point. So if it's above its isoelectric point, it'll be negatively charged at that point. So it'll probably be a -1, I want to say. Would it be a -2? Probably not. -2 would be that amino group also getting deprotonated. I'm going to go with -1. B. I can go back and check real quick, because I think they might have talked about glutamic acid specifically. Titration. Negatively charged under basic conditions. Yeah, I don't know. We don't need to go back. I'm pretty sure. Oh, actually, it might say specifically for 1. It has lysine for the positive case. For the acidic case, do they have an acid? An acidic amino acid. Yeah, they do talk about glutamic acid. At this point, it is electrically neutral. When it loses its proton, just as with glycine, its overall charge will be -1. Glutamic acid. Glycine or glutamic acid? Glutamic acid. When it loses its second proton, just with glycine, its overall charge will be -1. Great. -1. 3. Which of the following statements is most likely to be true of non-polar R groups in aqueous solution? Polar is hydrophilic, non-polar is hydrophobic. They are hydrophilic and found buried within proteins. They are hydrophilic and found on protein surfaces. They are hydrophobic and found buried within proteins. C, that sounds right. They are hydrophobic and found on surfaces. Hydrophobic matches with being buried. So we'll go with C. 3C. 4. Which of these statements concerning peptide bonds is false? Their formation involves a reaction between an amino group and a carboxyl group. Yes. They are the primary bonds that hold amino acids together. Yes. They have partial double bond character. Yes. Their formation involves hydration reactions. It's not it's dehydration, not hydration. So 4 is, or D is the one that is false. So that is the answer. 5. How many distinct tripeptides can be formed from one valine molecule, one alanine molecule, and one leucine molecule? How many distinct tripeptides? So order matters. If you reverse the order, it's not the same peptide because it has an N-terminus and a C-terminus. So valine, alanine, leucine. I'm just going to get these symbols real quick. Valine, alanine, leucine. Valine, V, alanine, A, leucine, L. VAL. All right. You could have VAL, VLA, AVL, ALV, and V in front, A in front, and L in front. LAV and LVA. Now VAL is not the same as LAV because there is an N terminus and a C terminus. And so they are distinct, but I think these 6 have to be it. 1, 2, 3, 4, 5, 6. There's not 27. I'm going to go with C, 6. Number 6. Which of these is most likely to be preserved when a protein is denatured? Quaternary structure, tertiary structure is going to get ruined. Primary and secondary are most likely to be okay. So I'm going to go with primary is the most likely to survive. Even hydrogen bonds will be disrupted, but I think the peptide bond isn't really going to get disrupted unless you have enzymes doing it. I don't think just heat is probably going to do that. 7. An alpha helix is most likely to be held together by disulfide bonds, hydrophobic effects, hydrogen bonds, ionic interactions between side chains. Well, we know the disulfide bond is up at a higher level here, and it makes loops. The alpha helix structure is due to hydrogen bonds between a carboxylic acid group and a amide group, amino group 4 peptides down, right? So that's the hydrogen bond. C. Number 8. Which of the following is least likely to cause denaturation of proteins? Heating the protein to 100 degrees Celsius, adding 8 molar urea, moving it to a more hypotonic environment, adding a detergent such as sodium dodecyl sulfate. Least likely to denature. Heat will denature. Both urea. Well, does urea denature? It does, doesn't it? Either way, urea and SDS will denature, moving it to a more hypotonic environment. hypertonic versus the hypotonic. Hyper means more salt. Hypo means less. Having lower osmotic pressure. So that would be least likely to denature proteins. It'll affect whether something like hemoglobin is picking up oxygen or dropping it off. In the hypotonic environment, it's probably going to, with respect to oxygen, it's probably going to let go of oxygen, but it's not going to denature the protein. I think that's within its normal functioning, going into hypotonic environments. So, 8C. 9. A particular alpha helix is known to cross the cell membrane. Which of these amino acids is most likely to be found in the transmembrane portion of the helix? We learned that was it not cysteine? Was it glutamate? Is often found in the transmembrane portion. So that's part of tertiary structure, or was that quaternary structure? Proline residues are rarely found in alpha helices, except in helices that cross the cell membrane. A particular alpha helix is known to cross the cell membrane. Which of these amino acids is most likely to be found in the transmembrane portion of the helix? Proline makes kinks. And so if you see it in an alpha helix, it's going to be at the cell membrane. But proline is not an option. But the reason for that is that proline has this ring structure that affects its flexibility, rigidity. So why don't we look for other molecules with rings? Proline, phenylalanine, glutamate is not a ring. So which of these amino acids is likely to be in the transmembrane portion? Well, the outside of the membrane is hydrophilic. The inside of the membrane is hydrophobic. So you want a hydrophobic, non-polar side chain. So proline, leucine, isoleucine, methionine, valine, alanine, glycine are none of the options. So none of those. Tryptophan, tyrosine, phenylalanine. Phenylalanine is in there. So that looks good so far. Polar. The polar ones would not. And none of these are listed anyways. Negatively charged, acidic. Aspartate, glutamate. Both negatively and positively charged are polar and thus can. Arginine is not an option. Lysine is an option. Histidine is not. Of these, I like phenylalanine because it's also that they talked about proline in the ring structure. Even though I don't know if this ring does quite the same for you as this ring. Negatively charged, both aspartic and glutamate. So I wouldn't choose between those two, so I'm just going to leave both of those. Lysine, positively charged, basic. But if negatively charged isn't, I wouldn't think positively charged would either. To me, aspartate, glutamate, and lysine, you could have equal footing. Phenylalanine is a little different. I'm going to go with phenylalanine, but we'll see what the answers say in a minute. 9, I'm going to go with C. We'll see how that goes. 10, which of these amino acids has a chiral carbon in its side chain? In its side chain. Serine, threonine, isoleucine. Chiral atoms in the side chain. Let's go one by one and check each of them out. Starting with serine. Side chain. This carbon has 2 hydrogens. That is not stereogenic. That is not chiral. Is it? If you can't tell the 2 hydrogens apart, you cannot say its side chain is chiral, so not serine. Threonine, its side chain is chiral, because this carbon is different than this carbon, is different than the OH, is different than the H that it's probably also attached to. Threonine is stereogenic, or chiral. Isoleucine, its side chain this isn't. This is chiral. It's got a hydrogen, it's got an alkyl group, it's got a larger alkyl group, and then it's got your alpha carbon. I think threonine and isoleucine both pass the test, 2 and 3, but not 1. I'm going to go with C for number 10. (11) Adding concentrated strong base to a solution containing an enzyme often reduces enzyme activity to 0, in addition to causing protein denaturation. Which of the following is another plausible reason for the loss of enzyme activity? Enzyme activity, once lost, cannot be recovered. B. The base can cleave peptide residues. C. Adding a base catalyzes protein polymerization. D. Adding a base tends to deprotonate amino acids on the surface of proteins. I'm going to go with that one because in addition to causing denaturation, they're looking for other reasons that it might not work. That enzyme activity, once lost, cannot be recovered doesn't explain an alternative reason for the protein not working. The base can cleave peptide residues. That sounds like denaturation as well. I mean, that's even more than denaturation. That's actually like lysing DNA, right? It's like C. Adding a base catalyzes protein polymerization. Protein polymerization is like making the protein, peptide bond formation. While it's not going to happen, it would denature the protein. You get a different protein in the end. Adding a base tends to deprotonate amino acids on the surface of proteins. That sounds like that's going to have an effect on its enzymatic activity. 11, I'm going to go with D. 12, which of these amino acids has a side chain that can become ionized in cells? Which side chain can become ionized? Let's run through them. Histidine. Can become ionized. So I think that's got to be it. Leucine does not. It's nonpolar, non-aromatic. Proline, nonpolar, non-aromatic. Threonine, while it's polar, Does its side chain become ionized? Yeah, if it gets deprotonated, it could get ionized. Does this get deprotonated before that? It does, yeah. This is from left to right, this is the first, followed by this. This is the second to get protonated. So from physiological pH going down, from 7 down to like 3 or 4, this will get deprotonated first. So maybe it's threonine, not histidine. We learned that histidine can become charged. We were told that it was histidine. It looks like threonine can also. So in cells, I guess the key word is in cells. At physiological pH, which of these is going to get charged? Since threonine is not in one of the charged cases, it's just polar, I'm going to say that at physiological pH, this doesn't happen, while at physiological pH, this probably does. I'm going to go with histidine A. 13, in lysine, the pKa of the side chain is about 10.5. Assuming the pKa of the carboxyl and amino groups are 2 and 9, respectively, the pi of lysine is closest to, so there's one at 2, one at 9, one at 10. The pi is going to be between 9 and 10. So 9.7, 9.8 sounds good. 13D. So this is heavily basic, so it'll have a high pi, right? Where's pi? Basic amino acids have high pi. And leucine, lysine, where are we? Lysine is basic. All right. 14. Which of the following is a reason for conjugating proteins? To direct their delivery to a particular organelle, to direct their delivery to the cell membrane, or to add a cofactor needed for their activity? To direct, isn't that more structure, tertiary structure, than quaternary structure? No, yeah, the lipoproteins, glycoproteins, nucleoproteins, that is to get them into their place, or is that just... These groups can also direct the protein to be delivered to a certain location, such as the cell membrane, nucleus, lysosome, or endoplasmic reticulum. So it is conjugated proteins. So I think all 1, 2, and 3, all are reasons to conjugate. So 14 will go with D. 15. Collagen consists of 3 helices with carbon backbones that are tightly wrapped around one another in a triple helix. Which of these amino acids is most likely to be found in the highest concentration in collagen? Well, we learned that... What is the backbone of the alpha helix? We learned that proline is only present in the beginning, so I think it's not going to be proline. Let's go back to where this helix part is. Proline makes a kink, so we're going to exclude proline. Glycine, threonine, cysteine. So we learned that the helix is made by hydrogen bonds between just the carboxylic acid and the amide, which is in all residues. But the disulfide bond is what's responsible for the two helices in a double helix to bond to each other. So I would imagine this triple would have a lot of those disulfide bonds. And the one responsible for disulfide bonds is cysteine. Not so much threonine, even though I think threonine also has... No, threonine doesn't. The methionine's also got a sulfur, but the cysteine is the sulfur that makes this disulfide bonds. So I'm going to go with cysteine. Whoops. 15, D. Now they said disulfide bonds are important. Did they say specifically that it's... They don't exactly say that the disulfide bond is between the two helices in a double helix, but I think that's the case. Disulfide bonds. I'm pretty sure that's the case. They didn't say that explicitly, but this triple helix idea has to have lots of disulfide bonds, I imagine. When they draw the double helix, right? Oh, this is going to be terrible. In between, they have these peptides that are linked. I think these are supposed to be, or are these just hydrogen bonds? Maybe they're just hydrogen. I thought these were actual disulfide bonds between the two helices of DNA. We'll find out in a second. 1 is B, 2 is B, 3 is C, 4 is D, 5 is C, 6 is A, 7 is C, 8 is C, 9 is C, 10 is C, 11 is B, not D. Bases can catalyze peptide bond hydrolysis. So choice A is incorrect. Enzyme activity can be discovered in at least some cases. Choice D is a true statement, but fails to explain the loss of enzyme activity. Let's look at 11 again. 11, adding concentrated base brings enzymatic activity to 0. In addition to causing denaturation, which of the following is a plausible reason? They said, I said D, adding a base tends to deprotonate amino acids on surface proteins. They said choice D is a true statement but fails to explain the loss of enzyme activity. On the other hand, choice A, enzyme activity once lost cannot be recovered, is incorrect. Enzyme activity can be recovered in at least some cases. So their only options are B and C. Adding a base catalyzes protein polymerization. They didn't have to explain that that doesn't seem to I guess that's not even a true statement. That's a nonsensical statement. B, the base can cleave peptide residues. A strong base can cause a hydrolysis reaction, I guess. That's something I guess we should have known. Is that what we're talking about? Is hydrolysis in the presence of a strong base? Hydrolysis can happen in both acidic and basic conditions. All right. 11b, 12a, 13d, 14d, and 15b, not D. Because collagen has a triple helix, the carbon backbone are very close together. Thus, steric hindrance is a potential problem. To reduce this hindrance, we need small side chains. Glycine has the smallest side chain of all, a hydrogen atom. So, not to do with the disulfide bond part, but just double helix is already pretty tight. A triple helix is too tight for large side chains, so we're just looking for the smallest side chain to fill it up. And there you go. Glycine, being the smallest of amino acids, is our natural choice. There you have it. That is peptides, amino acids, peptides, and proteins. Next time we'll be getting into enzymes, chapter 2.